everyone, welcome to Biology with MJ. This is MJ, your enthusiastic guide. So whether you are a NEET aspirant or simply a biology lover, this video is filled with valuable insights and knowledge. Okay, so stay tuned till the end of the video, and if you haven't already, please subscribe to my YouTube channel and press the bell icon so that you never miss an update. Remember to like, comment and share this video with your friends. Okay, so let's get to the video. Hello students, welcome back to my YouTube channel. This is the last lecture on the chapter Plant Kingdom. This is the lecture 5 of this chapter. It covers the topic Angiosperms and the plant life cycles and alternation of generations. So I know that these topics have been removed from the CBSE syllabus, but we have not received any such update specific to the NEET exam. So we will assume that we have to study everything for the NEET exam because that is a competitive exam and anything can come in that exam. Question from any topic can come. Unless the NEET has provided official notification about the removal of certain topics from the NEET syllabus, till then we cannot omit any topic. We have to cover everything. Okay. So in this chapter ends in this video. This is the last video of this chapter, and for the previous lectures you can check out the playlist attached here. Okay, I have attached the playlist link here. So the previous 4 lectures you can study from this playlist. Alright, so let us begin the video, and do not forget to subscribe to my YouTube channel and follow me on Instagram, Facebook and Telegram. Okay, don't worry, I'll remind you about this 2 to 3 times as the video follows. Okay, I'm sorry about that, but we have to do it as a YouTuber. So let's begin the video. So introducing angiosperms. What are angiosperms? Angiosperms are also known as flowering plants. As the name suggests, they produce flowers. They are the only groups of plants which produce flower, okay, which are responsible for the production of flowers. So sexual organ is the flower. Alright, so if we split up the term angiosperms, it is made up of these 2 words, angion and sperma. Now in gymnosperms we saw that gymnos means naked and sperma meant seeds. So gymnosperms are the group of plants which have naked seeds. That is, the seeds are not surrounded by a fruit wall, or the ovule is not surrounded by an ovary wall. But what happens in angiosperms? Angion means vessel and sperma means seeds. So angiosperms are those group of plants in which the seeds are surrounded by a fruit wall. That is, the seeds are contained in a vessel kind of structure. This vessel here refers to the ovary that surrounds the ovule. Okay, so if this is the ovule, okay, it is surrounded by ovary. So this ovule is surrounded by this ovary. This here is the ovary. Now this ovule will convert after fertilization. Post fertilization, ovule converts into seed and ovary turns into the fruit, right? So fruit here acts as the vessel for the seed. So this is how this term has been designated to these group of plants, because angion means vessel and sperma means seeds. So angiosperms are plants in which the seeds are covered or contained in a vessel. Okay, the seeds are not naked. So the seeds are formed inside the fruit. Okay, as already told, the seeds are formed inside the fruit. And the sporophylls are organized into flowers. Now in angiosperms, we studied in the previous lecture also. I gave you a knowledge point and indication about this specific information, that sporophylls in angiosperms organize themselves into structures called flowers. Whereas in gymnosperms, for example, if we talk about the specific group of gymnosperms, that is the conifers, In conifers, the sporophylls organize themselves into cones. Alright, but in angiosperms, the sporophylls are organized into flowers. In all the angiosperms, the sporophylls are organizing themselves into flowers. Okay. So as you can see, this is the image of a mango fruit. This here in the center is the seed and this is the fruit. Okay, this is the fruit wall and this is the seed. Previously, the seed was, how was the seed made? When the ovule post fertilization turned into seed. Okay. And how is the fruit wall or the fruit made? When the ovary or the ovary wall post fertilization turned into the fruit wall. So this is an introduction about angiosperms. This is the basic foundation which should be clear in your head in order to study further about angiosperms. Now, unlike the gymnosperms where the ovules are naked, in the angiosperms, which are also known as the flowering plants, the pollen grains and the ovules are developed in specialized structures called flowers. Now, ovules are a part of the female reproductive organ of a flower, and the pollen grains represent the male are the part of the male reproductive organ. All right. So both of these structures, they are formed in specialized structures called flowers. Both the male gametes or the male gametophytes and the female gametophytes are formed inside the flowers. And how are flowers formed? When sporophylls organize themselves into these specialized structures known as flowers. So in this slide on the leftmost image represents the flower. Now you can see certain structures inside the flower. Now these are the male reproductive organs which represent the stamen. Stamens are the male reproductive organ of the flower. We will study this in detail later on. Now these yellow colored powdery structures on the surface are pollen grains. Pollen grains represent the male gametophyte. And what are gametophytes? Which bear gametes. So these pollen grains bear the male gametes. Okay. In the center is present a structure which is known as the gynoecium. Okay. Now, if we look at a zoomed in view of this gynoecium, now gynoecium is a structure which is made up of one or more than one, if it is present in a group, one or more than one female reproductive organs known as the pistils or carpels. For example, these are stamens. A group of stamens will be known as androecium. Okay, this whorl together is known as androecium. Now, similarly, if a group of gynoecium, if a group of the female reproductive organ is present, female reproductive organ is the carpel. It is also known as a pistil. Now, if a group of carpel or pistil is present, it is known as gynoecium. If it is present in a single number, then also it is called gynoecium. But if it is present in more than one number, that whorl, that group is also referred to as the gynoecium. Okay. Now, let us look at the view of a single carpel or pistil. So, this structure in the middle represents the pistil. At the base of the pistil is present the ovary, and inside the ovary is present the ovule. The ovule can be present in a single number or multiple numbers. Okay, these circular structures which I am drawing are the ovules. Okay, so flower on the left, pollen grains in the middle are shown, and ovules in the ovary on the right is being depicted in this slide. So basically this slide is saying that the pollen grains and the ovules are developed in specialized structures known as flowers. Now you can see both these male and female reproductive structures are present inside this flower. All right, let's move forward. Now, angiosperms are an exceptionally large group of plants. They occur in a wide range of habitats because it is such a large group of plants. Okay, and the most evolved one also. If we look at how the plants evolved from Thallophyta, then bryophytes, pteridophytes, gymnosperms, and angiosperms. Angiosperms is the most evolved and most latest group of plants. Okay. Now, it varies in size also. We have the smallest angiosperm, which is known as the Wolffia. You can see on these small granule-like structures. A single granule represents the Wolffia plant. So you can see how tiny it is. Okay, it is the smallest angiosperm, Wolffia. And you can, on the other hand, on the extreme opposite are these tall trees of eucalyptus, which are some of the tallest trees that are present. Okay. Eucalyptus here should be italicized, okay? My mistake. Please consider this name in italics. It can grow over 100 meters tall. So, these trees are so large, their height goes over 100 meters. So you can see they are very diverse. Angiosperm as a group is very diverse. Now, if we talk about their economic importance, okay, if we talk about their economic importance, angiosperms are very useful to the mankind because they provide us with food, they provide us with fodder, okay, they provide us with fuel, with medicines and several other commercially important products. In all areas of our life, we extract something or the other from the angiosperms, be it either food or fodder. Fodder is the food product which we feed to the livestock, to the cattle. Then we obtain fuel from angiosperms. They are useful in medicines. Okay, there are certain herbs which have medicinal purposes and are also incorporated in medicines. And various other commercially important products are extracted from these angiosperms. Even wood is used in order to construct certain furnitures. Okay. In house, in the construction, wood is one of the most used product from the plants. All right. So this was about economic importance. Now, how is sexual reproduction carried out in angiosperms? Now, angiosperms are sexually reproducing plants. Okay. How does the sexual reproduction occur in angiosperms? Via the flowers. Flowers carry out sexual reproduction in angiosperms. Okay, flower is one of the most important and the defining organ of angiosperms. Now, if flowers are performing sexual reproduction, they have the male and female reproductive structures, that is understood. Okay, we talked about this in the previous few slides also, that flowers contain the male sex organ and the female sex organ. The male sex organ is the stamen and the female sex organ is the carpel. Now, if we dissect the structure of a stamen, it consists of anther and filament. Now, see, this long tapering structure is the filament and at the head is present this lobed structure, anther. If we consider the central structure, it represents the female sex organ, which is also known as the carpel or a pistil. It is made up of a top known as stigma. This structure here is the stigma. This central elongated portion is the style. This bulbous region, swollen base, represents the ovary, and these circular units are known as the ovules. Now, these are the ovules which will turn later into the seeds. Okay, and this ovary right here will turn into the fruit wall, into the fruit. So this was about the male sex organ and female sex organ of the flower. We'll study about these structures in detail in the further slides. Now, these colorful structures are the most attractive feature of the flower, which attracts pollinators to them, helps in pollination, dispersal of seeds. Dispersal of seeds happens later on. When seeds are being dispersed, their flowers do not play a role. Flower has already, their petals have been shed off and the seeds are being dispersed. But when pollination is happening, flowers needs some of their attractive features to attract the insects. Okay, what is that attractive feature? So that attractive feature is the presence of these colorful petals. So, okay, so these colorful structures are known as petals, which attracts the pollinators to the flowers. These are the sepals, these green colored structures. So the outermost whorl is this whorl of sepals. Then comes the whorl of petals. Then comes the whorl of stamens and in the middle is present carpel. Carpel can be present in a single number or in more than one number. Okay, more than one carpel can be present. Now let's move forward. Now let us look at the structure of a stamen. See, this is the anther. The top bilobed structure here represents the anther. This long structure is known as the filament. Each stamen consists of a slender filament with an anther at the tip. See, this is the anther and this is the filament. Filament helps the stamen to attach to the flower, and the anther bears what? Anther bears the pollen grains. Pollen grains are formed in the anther. Okay. Now coming to pistil, also known as carpel. This is the structure of a carpel. Topmost area is the stigma. Stigma is the area where the pollen grains will land after pollination happens. That is the transfer of the pollen grains to the female reproductive structure of a flower, which is the pistil or a carpel. The pollen grain first gets deposited on the sticky top surface, which is known as stigma. It is very sticky because it has certain secretions on its surface which is very sticky, and whenever a pollen lands on it, the pollen grain gets stuck to the surface of the stigma. This long structure is the style. This is the ovary wall and ovule, as already explained in the previous slide. Now the female sex organ in a flower is the pistil or the carpel. The pistil consists of an ovary which encloses one to many ovules. Now ovule can be one in number or more than one in number. Okay. Now pollination. Pollen grains, after they are dispersed from the anthers, see, these are the pollen grains. After they are released from the anthers, these structures, they are carried by the wind or various other agencies. The pollinating agent could either be wind, it could be water, it could be insects, okay, animals. So pollen grains, after they are dispersed from the anthers, they are carried by wind or various other agencies. Here a pollinator, insect pollinator is being represented, but it could be either wind also, it could be water also, or any other pollinating agent. Okay, so these are being carried via the pollinator to the stigma of the pistil. See, this is the pistil that is present. So this pollen grains are carried to the stigma of the pistil via the pollinating agents, which could be insects, wind, water, etc. And this event, this transfer of the pollen grains from the anther to the stigma of the pistil is referred to as pollination. This event is known as pollination. Okay. Now flowers can be of two types. If in a flower both the male and female reproductive structures are present together, that flower is known as a monoecious flower, okay, a bisexual flower. Now there are certain flowers in which either only the stamens are present or only the pistil are present, okay, and that is known as a dioecious flower or a unisexual flower, okay, because only a single sexual structure is present in it. So there is also a lot of theory that goes into it, but for now you need to know this much only as per the information that NCERT has provided. In the sexual reproduction of flowering plants, which is the chapter in class 12th, we will study all about the angiosperms in further details. Now before moving forward, do not forget to subscribe to my YouTube channel, follow me on platforms such as Instagram, Facebook and Telegram. I have the presence of this channel on all of these three platforms, and the links to these platforms are provided in the description of this YouTube video. So you can check out the description, click on the links to these platforms, and please, you will be helping me a lot if you follow me on these platforms, because it will help me to reach the maximum number of students. Okay, so please kindly do that. It would mean a lot to me. And let's move forward now. So if we are dividing angiosperms, angiosperms can be divided into two groups, monocots and dicots. Now the term monocots means, mono means one and cots represent cotyledon. So if the seed has only one cotyledon, that plant is known as a monocot. If the leaves of that plant have parallel veins like this, okay, straight veins that are running parallel to each other. That is also a defining feature of the monocot. If their flowers are present in the multiples of 3, if the whorls in those flowers are present in the multiple of 3, for example, sepals and petals either comprise 3 petals. For example, if we talk about petals, if the petals are 3 or 6 or 9, if such in the multiples of 3 the whorls are present in the flower, that is also a defining feature of monocots. Alright, now talking about dicots, in dicots the seeds have 2 cotyledons, because di means 2 and cots means cotyledons. So in dicot plants the seeds have 2 cotyledons. If we look at their leaves have branched veins. See, here the veins were straight running parallel to each other, but here the veins are branched and further divided into more branches. Veins are present and each vein is further dividing into more veinlets. Alright, now if we talk about their flowers, the whorls in the flowers are present in the multiples of 4 or 5. The flowers, that is, they can be tetramerous or pentamerous. Here the flowers are trimerous, that is present in the multiples of 3. Now here these petals could be present in the multiples of petals, or sepals could be present in the multiples of 4 or 5. For example, 4, 8, like that. Okay, so this was the major difference between monocots and dicot plants. Now, what are cotyledons? Cotyledons are a part of the embryo. Now, har ek seed mein embryo will be present. All seeds would have an embryo, right? That is why they are germinating in order to form a new plant, because this embryo is converting into this new plant. It is responsible for the formation of this new plant. Embryo itself is the future plant in its very young stage. Now, each embryo will have cotyledon. Cotyledons are the part of the embryo which are responsible for the production of the first leaves when the seed germinates. Okay, and the initial source of nutrition is also provided by these cotyledons to the developing plant. Now, in monocots, only a single cotyledon is present. Okay, and in dicots, 2 cotyledons are present. So, this structure is the embryo. Here, this structure is the embryo. So, 2 cotyledons are present in dicots. An example of a dicot is bean. Then monocot example is corn plant or maize. Okay, it has 1 cotyledon in its embryo. So this was about the difference between dicot and monocot plants regarding their seeds. So this is the image of a dicot plant which is given in the NCERT, and this is the monocot plant. Okay, see, as you can see, the leaves have parallel kind of venation. It is not very clear in this image, but if we will zoom it, the veins in these plants are running parallel to each other. Then this is the dicot plant, in which each leaf has branched veins. Okay, so you can spot these differences. You can go around in your neighborhood, in the plants that you have in your home. You can just go and observe as a homework. You can go and observe which is a monocot plant, which is a dicot plant. Now, monocots are usually, grasses are monocots, because all of the grasses have all these features, that is parallel veins. You can just go outside in your home garden or in your neighborhood park and observe, distinguish between what is a dicot plant and what is a monocot plant. I highly recommend this activity to you because it will further clear your concepts up. Now, if we talk about reproduction in angiosperms, let's talk about the male reproductive organ first. So we have already studied about the stamen. Now, if we cut a transverse section of the stamen, if we cut the stamen, particularly the anther, like this, and observe the structure, in the center of these are the 4 sporangia that are present, because the anther is a bilobed and the tetrasporangiate structure. So, if we dissect the anther, these 4 sporangia can be seen, not lobes. This is a lobe and this is a lobe. That is why it is known as the bilobe structure. But each lobe has these 2 sporangia. So inside each sporangia, these pollen mother cells are present, which undergo meiosis. Now, pollen mother cells are also known as the microsporocyte. Okay,
when they undergo meiosis, these are diploid. Okay, when they undergo meiosis, they form a tetrad of microspores. After meiosis, the cells that are formed, they are haploid. Okay, so we are writing N here. So these are the tetrad of microspores that is formed. Now each microspore will develop into a pollen grain. Okay, each of these microspore will develop into pollen grain. So here microgametogenesis is written because it is the formation of the male gametes. Pollen grain is the male gametophyte which bears the male gametes. So the male gametophyte is the pollen grain. Gametophyte is a structure which bears the male gametes. Okay, so here the male gametophyte is the pollen grain. So these microspores will develop into the pollen grain like this. So each of this microspore, if we look at it, it will become polarized, formation of vacuole will take place, then mitosis 1 will occur, leading to the formation of a vegetative cell and a generative cell. Then if now two cells are formed in this microspore, one vegetative cell and one generative cell, that is why it is known as a bicellular pollen. Okay, and the see numerous vesicles. Now here a single vacuole was present, now numerous vesicles are present here. Now this bicellular pollen will undergo mitosis 2 in order to form a tricellular pollen. In the tricellular pollen, two sperm cells are present. So the generative cell undergoes mitosis 2 in order to form two male gametes. These two purple colored structures are the two sperm cells or the male gametes. These are formed from the generative cell. Okay. Now this stage is known as the tricellular pollen because now three cells are present. Okay, one vegetative cell and two sperm cells. This was known as the bicellular pollen because here two cells are present, one generative cell and one vegetative cell. Now pollen grains can be shed at any of these two stages. Okay, either they could be shed at the bicellular stage or they could be shed at the tricellular stage. The pollen grains which are shed at the bicellular stage later on, once they land on the stigma of the flower and they grow into the pollen tube, they convert into the tricellular stage by undergoing mitosis 2. So and sometimes the mitosis 2 happens later after pollination has happened, and sometimes even before the pollen grains are shed, the mitosis 2 has already happened and the pollen grains exist in that tricellular state. So after Ncert dehiscence, that is the release of pollen grains, pollination will take place and the pollen grains will land onto the carpel, stigma of the carpel or the pistil. So this was about the development of the male gametophyte. Now talking about the development of female gametophyte. Now female gametophyte refers to the embryo sac. And embryo sac bears the egg, which is the female gamete. So the female gametophyte is the embryo sac and gametophyte bears the female gamete, which is known as the egg. Okay. Now this is the pistil or a carpel. This structure here is the ovary. If we zoom this structure, we can see this is the ovary which contains numerous ovules. See, these structures are the ovules. If we zoom in one ovule, we can see that this bears an embryo sac. This structure here is the embryo sac. And embryo sac bears what? Embryo sac bears the egg cell. This is the egg cell, which is also known as the female gamete. So this is how the development of egg cell takes place. This is the order it is following, and this is how it is. The female gametophyte or the embryo sac is arranged inside the ovary or inside the ovule. In this image, the outside structure is the ovule. Okay, this embryo sac is present inside the ovule. This yellow colored structure and everything beyond this structure represents the ovule. This is the embryo sac, the central, the circular portion here. This is the embryo sac. Rest everything is the part of the ovule. And embryo sac is also part of the ovule, but it is formed inside the ovule. Now let's talk about the the development of female gametophyte. So there is ovule that is present. Now, if we are saying that the embryo sac is formed inside the ovule, how is it forming? There must be something inside the ovule which is converting into the embryo sac, right? So just like in the development of male gametophyte, pollen mother cells were present, or the microsporocytes were present in the female gametophyte development also inside the ovule or the megasporangia. Now, ovule is also known as a megasporangium. Okay, here these structures are the microsporangia. Okay, each of these structures is known as the microsporangia. In the microsporangia, microsporocytes are formed, microsporocytes are present which are undergoing meiosis. Similarly, in the development of female gametophyte also, ovule represents the megasporangium. Megasporangium is the singular term and multiple ovules are referred to as megasporangia. So each of the megasporangium will bear megasporocyte, right? Megasporocyte or the megaspore mother cell. It will undergo meiosis. See, this is the megaspore mother cell which is diploid. It is present inside the ovule. It will undergo meiosis in order to form 4 haploid cell or the tetrad of megaspores. Now, one very unique thing happens in the development of female gametophyte. Now, in the development of the male gametophyte, all of the microspores, the tetrad of microspores, were converting into pollen grains, were developing into pollen grains. But in the female gametophyte development, only one megaspore remains functional and the rest 3 megaspores degenerate. Okay? Only one remains functional. So as you can see in this image, these are the tetrad of megaspores. One of the megaspores will grow further and the rest 3 degenerate. Now this megaspore, it will develop into what structure? This megaspore will develop into this mature embryo sac. And this is the procedure it is following in order to do that. Okay? See what is happening. This megaspore is maturing, increasing in size, then it will undergo meiosis. Okay? Mitosis. In mitosis, what is happening? The nuclei is first dividing via the mitosis, then cells are formed. Ultimately, a mature embryo sac, the structure which we call as the mature embryo sac is formed, and in the cellular and the nuclear arrangement in a mature embryo sac looks like this. Egg cell is present or the female gamete is present inside the mature embryo sac. So via the mitosis, see how many mitoses are happening? 1, 2, 3. After mitosis 1, 2 nuclei are formed. After mitosis 2, 4 nuclei are formed, and after mitosis 3, 8 nuclei are formed. So 8 nuclei are formed and they are arranged into 7 cells. Now you must be wondering why 7 cells? See, 1 cell here, 1 cell here, 1 cell here. 3 cells, 4, 5, 6, 3 cells at the bottom. Now in the center also is present a cell which is known as the central cell. So this central area here is representing the central cell. Now this central cell is binucleate. It contains 2 polar nuclei. That is why the condition is 7-celled and 8 nucleate. Now here what this process, the formation of the from the megaspore, this process from right here to here, this is megagametogenesis. And this formation of the megaspore up till here from the megaspore mother cell is the megasporogenesis. Just like we studied in the development of the male gametophyte, this procedure from here to here, from the pollen mother cells or the microsporocytes, the development of the tetrad of microspores from the pollen mother cells is the microsporogenesis, because these are the microspores that are formed. From microspores onwards, the development of male gametophytes take place, or the pollen grains takes place, which bear the male gametes. So this procedure from this microspore to the development of this tricellular pollen is known as the microgametogenesis, because in this procedure the male gametes are forming. Okay, and each of these microspores will undergo microgametogenesis like this. So each of these microspore will become polarized, will undergo mitosis, will form certain vesicles, a number of vesicles, and will form the bicellular and ultimately the tricellular pollen. So this megaspore is finally germinating into the mature embryo sac. Okay, so megasporocyte here was diploid. The tetrad of microspores is haploid. One remains functional, which is haploid. Okay. Now this one functional megaspore is undergoing mitosis only to form the mature embryo sac or the female gametophyte. So the embryo sac in its ploidy level, no matter how many number of cells it comprises, all of these cells are haploid only. Okay. Only the central cell comprises of 2 nuclei, but each of those nuclei are also haploid. Okay. N plus N. This nuclei is also N, this nuclei is also N. Okay, N plus N condition is present in the central cell. So the embryo sac is haploid only, and the egg cell, which is the female gamete, is obviously a haploid structure because it is formed from a haploid structure via the mitosis, and that haploid structure was the megaspore. Now this is the detailed structure of an embryo sac. Each embryo sac, as I've already told, comprises of a 3-celled egg apparatus. Now you can see here, this is the egg cell. Okay, let me choose a different color here. This is the egg cell, and the egg cell has along with 2 synergids it is present. Okay, 2 synergids are present, which are present along with the egg cell at the micropilar end. Micropilar end is the one from where the pollen tube will enter. This image, yeah, in this image, this is the ovule. You can see there is an opening in this ovule. Wherever this opening is present, that end is known as the micropilar end, and the end opposite to that here, this end is known as the chalazal end. Chalazal end. Okay, so this micropilar end here bears the egg apparatus. This is the egg cell, which is surrounded with 2 synergids, and this represents the egg apparatus. 1 cell and 2 synergids comprise the egg apparatus. Egg cell and synergids comprise the egg apparatus. Okay, this here. So egg apparatus is 3-celled. It bears 3 cells, 1 egg and 2 synergids. Now opposite to the micropilar end is present the chalazal end, which bears 3 antipodal cells. See, these 3 cells here are the antipodals. In the center is present this cell, which is known as the central cell. See, the central cell is present in the center, which bears 2 polar nuclei, 1 nuclei and the second nuclei. Now, initially they are not present they have not fused with each other, they are present separately. But eventually these will fuse in order to form a diploid secondary nucleus, that is, they will form a 2n condition. Initially they are present in n plus n condition. When they are not fused, when they will fuse, eventually they will fuse and they will form the 2n nucleus or the diploid secondary nucleus. But each of the other cell here is haploid only. Okay, n, n, n. Synergids are also n, egg cell is also n, and these polar nuclei are also n until they have not fused with each other. When they fuse with each other, they will become diploid. They will form a single nucleus, which is diploid. Okay, so this is the structure of a embryo sac. And how many cells does it have? It has 1, 2, 3, 4, 5, 6, 7. 7 cells, and number of nuclei are as many as the number of cells plus 1 additional nucleus, because the central cell has 2 nuclei. So 8 nuclei are present. So 7-celled and 8 nucleate condition is present in the mature embryo sac. Now for fertilization process in angiosperms, they need a mature embryo sac, number 1 requirement. And the second requirement is the presence of a pollen tube with male gametes. Okay, what is fertilization? Fertilization is the fusion of the male and female gametes, right? That is the fusion of the male gamete with the egg cell. So what would be the requirements for it? Obviously the presence of a mature embryo sac which has a egg cell, and the second requirement would be the presence of male gametes, and male gametes are delivered how? Up to the micropylar end via the formation of the pollen tube. Okay, the pollen grain, when it lands on the stigmatic surface, will germinate. Okay, the vegetative cell will germinate and form this pollen tube. Inside the pollen tube are present these two male gametes and one tube nucleus. This tube nucleus will degenerate. Later on in the process, it will degenerate, and only these two male gametes will enter the micropylar end and are delivered to the egg cell. One of these will fuse with the egg cell, and the other will fuse with these polar nuclei. We will study about this process later on, but for now just understand that in order for the male and female gametes to fuse with each other, we need number one, a mature embryo sac, and second, the presence of a pollen tube with these male gametes. Now the pollen grains, as already told, they germinate on the stigma, and the resulting pollen tube, see this pollen tube forms. Now we know that the female reproductive structure is the carpel, which looks like this. So whenever the pollen grains lands onto the surface of the stigma, it will germinate to form a tube-like structured pollen tube, which will grow like this. In the ovary is present let me choose a different color here. In the ovary, ovule is present like this with an opening. And inside the ovule is present what? The embryo sac. And embryo sac has what? The egg cell, right? So the pollen tube which bears the two male gametes will enter the ovule via the micropylar end, then it will enter the embryo sac and it will reach the egg cell. And what does the pollen tube has? Pollen tube has the two male gametes. One of these will fuse with the nucleus of one of these male gametes will fuse with the egg cell, and the other will fuse with the central cell, with the two polar nuclei of the central cell, not the central cell, with the polar nuclei of the central cell. So in this statement also the same is written, that the pollen grains germinate on the stigma, the resulting pollen tubes grow through the tissues of the stigma and the style, and they reach the ovule like this. See, this is the stigmatic surface, this is the pollen tube, these are the male gametes, this is the tube nucleus. Tube nucleus is the nucleus of the vegetative cell, and these two male gametes, they are formed when the generative cell has divided via mitosis 2 into two male gametes. So this tube nucleus, as already told, will later on degenerate. You have a graphic representation, a better picture here. See, this is the pollen. Okay, this is the pollen tube that has formed. It comprises of the generative nucleus. Now see, only a single nucleus is shown here because this is showing a bicellular pollen. Okay, see, the tube nucleus is growing towards the micropylar end. So when the pollen tube has finally reached the micropylar end, the tube nucleus degenerates. Okay, so basically the tube nucleus is guiding the pollen tube towards the micropylar end. These are the two sperm nuclei that are present, because ultimately the generative nucleus has divided into two male gametes. Okay, hence two sperm nuclei are formed. These represent the two male gametes. See, ultimately the bicellular pollen has reached the tricellular state here. Okay, ultimately it has divided into two male gametes only, and it will happen in all kinds of pollen grains, be it a bicellular pollen or a tricellular pollen has already the two male gametes. But in a bicellular pollen, this division, this mitosis 2 will happen later on in this stage when the pollen tube is about to reach the embryo sac. So here it finally enters the micropylar end here, and it will the sperm nuclei will then undergo the fertilization process. We will study about this process now. Now, since there are two male gametes, right, two male nuclei we studied, two male gametes are present. Now, one of these male gametes, see these are the two male gametes or the two sperm cells we can also say. One of these male gametes will fuse with the egg cell. This cell here represents the egg cell. So, one of these male gametes is fusing with the egg cell to form a zygote. Fertilization is happening here. This process is known as syngamy. Okay, when one male gamete fuses with the egg cell to form a zygote, that is N plus N is equal to 2N. Okay, N is the egg cell, the other N is the sperm cell. They fuse to form a zygote or the 2N cell. This process is the syngamy process. Now, syngamy has happened. What about the other male nuclei? The other male nuclei goes and fuses with the two polar nuclei of the central cell in order to form a triploid primary endosperm nucleus. Now, when this male gamete, when it will fuse with these two polar nuclei will already have fused to form a diploid secondary nucleus. Even if initially they are present as two separate nuclei, they would have eventually fused together to form a diploid secondary nucleus before the second male gamete reaches towards them. So, when the second male gamete reaches to them, they form this triploid primary endosperm nucleus, okay, which is also known as PEN in its short form. Why triploid? Because its ploidy level is 3N. Okay. Now, this 3N nucleus, it is responsible for the production of the endosperm. Endosperm is the tissue which provides nutrition to the developing embryo, and the embryo is formed from the zygote. So, syngamy plus triple fusion represent the process of double fertilization. Why? Because two events are happening here. Two fertilization processes are happening. One is syngamy, that is the fusion of male and female gamete, which is the syngamy, fertilization number 1. Second fertilization process is the triple fusion, where the second male nuclei or the male gamete is fusing with the diploid endosperm, with the diploid secondary nucleus. Okay, when it is fusing with the diploid secondary nucleus, that is the triple fusion process. Since both of these are fertilization processes only. So, two fertilizations are happening. That is why this process is known as double fertilization, and this event is unique to angiosperms. Okay, in no other plant group this event is seen. It is exclusive and unique to angiosperms. Now, let's move forward. So, what happens post fertilization? Now, fertilization has taken place. Post double fertilization, what are the events that take place? Now, see, post double fertilization, this is how the embryo sac will look like. The zygote has turned into the embryo, okay, because the zygote has formed. Now it will divide further in order to form the embryo via the mitosis. The zygote forms the embryo, which is the future plant. Okay, see, this is the zygote. It is now dividing itself into the embryo. The primary endosperm nucleus is present. See, this is a single nucleus now. Initially, 3 nuclei were present, out of which 2 nuclei were of the central cell, which fused together in order to form a diploid secondary nucleus before fertilization took place. When the second male gamete arrived, this diploid secondary nucleus fused with the second male gamete in order to form the primary endosperm nucleus, which is triploid, that is its ploidy level is 3N. Now this is a single nucleus which has a 3N ploidy level. It is present inside the central cell, which is now known as the primary endosperm cell, because this cell will convert into the endosperm. It will divide into the endosperm tissue, and endosperm tissue provides nourishment to the developing embryo. It is a nutritive tissue which provides nutrition to the developing embryo, because embryo will need certain nourishment and nutrition in order to grow. Where does this nourishment come from? This nourishment comes from the endosperm. So, endosperm provides nutrition to the developing embryo, and when the seeds are dispersed, how will the embryo get sustained until okay, there is a dispersal mechanism that is happening. Okay, up until the seed is not getting a favorable environment to germinate, till then there needs to be a source of nutrition for the embryo to sustain itself, to be alive up till it has found a suitable environment or a favorable condition to grow. So up till then, this nutrition is provided by the endosperm tissue. The central cell, as already told, has converted into the primary endosperm cell because it now contains the primary endosperm nucleus. So automatically it is now called as the primary endosperm cell. Okay. The synergids, they degenerate. So these two synergids which were present, they will degenerate. The antipodals here will also degenerate. The ovule, okay, the ovule inside which the embryo sac is present, this is the embryo sac. The ovule will turn into seeds. The integuments will harden. The ovules, the surface of the ovules, the outermost layer of the ovules comprises the integuments, layers known as integuments. They will harden themselves into seed coats and ultimately form a structure which are known as seeds. The ovules are further surrounded by the ovary, so the ovary will turn into the fruit, which is the fleshy part which we consume whenever we eat a fruit. Now this embryo which is formed, it can either have one or two cotyledons depending upon whether it is a monocot plant or it is a dicot plant. Okay. Now we talked about the formation of endosperm from the primary endosperm nucleus. So how is the endosperm formed? So this primary endosperm nucleus will undergo multiple divisions without cytokinesis. Okay, so this this nucleus will undergo karyokinesis, that the nucleus will divide mitotically into multiple nuclei without cytokinesis, resulting in the formation of a multinucleate endosperm. Okay, this is the most common type of endosperm that is formed, the nuclear type of endosperm, in which the cytokinesis is not following the karyokinesis, that the cell division is not happening as the nuclei are dividing. Cell division will occur later on in the later stages, then multicellular endosperm will be formed. But then there is also a second kind of endosperm formation which is known as the cellular endosperm formation. In cellular endosperm formation, karyokinesis is followed by cytokinesis from the initial stage only. Okay, whereas there is a third type of endosperm also which is known as the halobial endosperm, which is an intermediate between the nuclear and the cellular endosperm. But all this information about endosperms is additional information which you need not remember for this chapter. Okay, this is just an additional piece of information for your better understanding. But this is how endosperm is formed from the primary endosperm nucleus. And the nuclear endosperm is the most common type of endosperm formation method that is seen. Now let us look at the life cycle of angiosperms. So the flowering plant or the plant that we see, it is a sporophyte. As we move from thallophytes or algae to bryophytes, then pteridophytes, then gymnosperms, then angiosperms, what is happening? Gametophyte is becoming reduced and more dependent on the sporophyte. Sporophyte is becoming more and more independent and the dominant plant body. Initially, gametophyte was independent and sporophyte was dependent. Gametophyte was the dominant plant body, it was bigger in size, and sporophyte was reduced. But as we move further through the plant groups, as the plant groups evolved into angiosperms, slowly and gradually, as we saw in all these plant groups, the gametophyte started reducing in size and it became dependent upon the sporophyte. The sporophyte grew in size, became a more dominant structure, okay, and it became independent. Okay, so the flowering plant, we see that the flowering plant body that we see in angiosperms, all we see is the sporophyte. Okay, the gametophyte stage is very reduced, as we saw. Pollen grains is the gametophytic stage. It is so reduced and its lifespan is also very short. Okay, after it is released, it will fuse with it will go to the stigma, then it will reach the female gamete, it will fuse to form a zygote. Again, whenever the zygote is formed, again the sporophytic stage comes into the picture. And female gametophyte is the embryo sac. It is also short-lived. As long as fertilization has not taken place, only up till then they are existing. As soon as the fertilization occurs, again the sporophytic stage comes into the picture. So this flowering plant represents the sporophytic stage. This is the dominant stage. Sporophytic generation is 2n, diploid, and gametophytic generation is n, haploid. Now this sporophytic generation, it will form the gametophytic generation, right? So how it is forming the gametophytic generation? See, it contains the male and female reproductive organs. This is the female reproductive organ, which is the carpel. This is the male reproductive organ, which is the stamen. They comprise the microsporangium and the megasporangium respectively. Microsporangium will comprise the microspore mother cell, and the megasporangium will comprise the megaspore mother cell. The megaspore mother cell is diploid. Similarly, microspore mother cell is also diploid. So up until here, the sporophytic generation is seen. But when these respective cells will undergo meiosis, when the megaspore mother cell and microspore mother cell will undergo meiosis and form the tetrad of microspores and megaspores, then the gametophytic generation begins. As soon as the end ploidy level comes into the picture, we can say that the gametophytic generation has begun. So the microspores will develop, each of these microspores will develop into the pollen grains. Okay, and the tetrad of megaspores that was formed, out of these tetrad of megaspores, what happened? What did we study? 3 of them will degenerate, only one of the megaspore will remain functional and will form the embryo sac, which contains the female gamete, that is known as the egg cell. Okay, this pollen grain also will bear 2 male gametes. See, these are the 2 male gametes. So when this male gamete will fuse with the female gamete, it will result in the formation of a zygote. Again, the sporophytic generation began. So as you can see, the color changed. Okay, this was dark yellow color, this was light yellow color. All of the plants, all of the elements or the parts of this life cycle of this plant that are falling in the light yellow zone represents the sporophytic generation, and the elements that are lying in the dark yellow part represents the gametophytic generation. So the zygote is 2n. It will form the embryo, which is also 2n, because zygote forms the embryo via the process of mitosis. And then embryo will form the sporophytic plant body. Okay, or it will form the sporophyte. So as you can see, the sporophytic generation is dominant. In this whole life cycle, the majority of the part is being occupied by the sporophytic generation. So the sporophyte is dominant. Now you can also see this is free living. It has its own method of production of food because it has green leaves which will perform photosynthesis, will form food for this plant. It has a proper well-developed root system. It is independent in existence. So the sporophyte is dominant, independent, and independent means it is free living. Okay, it is not dependent upon the gametophyte. But now let's come to the gametophytic generation. Gametophytic generation is highly reduced and it is dependent on the sporophyte in order to survive. It cannot survive on its own. It needs the nutrition from the sporophytic plant body in order to form, okay, and then survive. Okay, so this was about the life cycle of angiosperms. A more detailed diagram is seen in this image. See, this is the flower. Here microsporogenesis is taking place, then microgametogenesis is taking place, resulting in the formation of 2 sperm cells. Similarly, in the female reproductive organ, megasporogenesis is taking place, then megagametogenesis is taking place, resulting in the formation of the mature embryo sac. Here, the 2 sperm cells are forming. So this is the gametophytic stage in both the development of male gametophyte and in the development of the female gametophyte. So ultimately, fusion will occur of the male and female gametes will fuse with each other, result in the formation of a zygote, then which is the syngamy. The second fertilization will happen with the diploid secondary nucleus in order to form the triploid endosperm nucleus, which will form the endosperm. Then seed will form because the ovule will turn into the seed and the ovary will turn into the fruit. So the seed would be surrounded by the fruit. So whenever the seed will germinate, whenever it finds a suitable environment to germinate, it will form the young sporophyte. Okay, S should be written here. So young sporophyte is formed, which will mature into a mature plant, adult plant, or a mature sporophyte. So this was about the life cycle of angiosperms. And double fertilization, we saw that it is happening during the process of during the life cycle of angiosperms. During the fertilization, two fertilizations are happening. So very, very important. Double fertilization is a unique feature of angiosperms, and a lot of questions appear from this topic. Do remember it. Now let's study about plant life cycles and alternation of generations. So in all the plant groups that we have studied, we saw that haploid cells were also present, diploid cells were also present, and both of these cells could divide by mitosis in order to form different kinds of plant bodies. Haploid cells form the haploid plant body, diploid cells form the diploid plant body. If they are dividing by mitosis, they would form something or a structure which has the same ploidy level, right? Because mitosis is equational division. The ploidy level remains the same in the progeny cells. So the haploid, if we look in general at the life cycle of any plant, the haploid plant body represents the gametophytic stage because it has n ploidy level, n number of chromosomes, one set of chromosomes is only present. Now the gametophytic stage will form what? Gametes, it will be your gametes, right? And gametes also have the n ploidy level, right? So यहाँ पे mitosis होगा या meiosis होगा in order to form the gametes. So if the ploidy level is being sustained, then mitosis is occurring. So gametophyte undergoes mitosis in order to form gametes. The male gamete and female gamete will then fuse with each other in order to form a diploid zygote. Now the diploid zygote represents the sporophytic stage, right? Now the sporophytic stage represents the diploid plant body. The zygote will form embryo and the mature sporophyte. Now zygote is 2n, the mature sporophyte is also 2n. Mature sporophyte is the diploid plant body. So in order to form the sporophyte, see again the ploidy level is being maintained. So what will happen? Meiosis or mitosis? Again mitosis will happen. Okay, so zygote will divide by mitosis to form the sporophyte. Now the sporophyte will again form spores. And how are the spores forming? Now spores have the ploidy level of n. Because these spores, they have to give rise to the haploid plant body. So sporophyte undergoes meiosis in order to form spores because the ploidy level has to be halved in the spores. So in order to halve the ploidy level, reductional division has to take place, which is meiosis. Now these spores, why their ploidy level had to be reduced or halved? Because they will divide by mitosis in order to form the gametophyte or the haploid plant body. So the sporophyte produce spores via meiosis, and the spores, whenever they find a suitable environment, they germinate via mitosis in order to form the gametophyte or the haploid plant body. So this is how the generation, see this spore box should also be in the same color as this one, okay. Mistakenly I did not change its color, but it should be in a yellow color. It is my mistake. So you can see here two generations are present. If we can divide it like this. Two generations are present. Generation number 1 and generation number 2. So all of these yellow colored structures represent the haploid generation or the gametophyte generation with the ploidy level n. And all of these purple colored structures represent the diploid generation or the 2n generation with the ploidy level 2n, double the number of chromosomes. So this is the sporophytic generation in purple and this is the gametophytic generation. So all of these plant groups whichever we have studied so far have shown these alternation of generation. So this alternation of generation is seen in the life cycles of all the plants. However, in different plant groups as well as the members that are present in them, there are certain variations. Okay, the life cycle, the overall pattern is we have understood from what we have understood is that there is an alternation of generations. That is, haploid generation is alternating with the diploid generation. Gametophytic generation is alternating with the sporophytic generation. But the duration of these generations is different depending upon the plant groups or depending upon the members of what members of the plant groups we are talking about. So based on these variations, different patterns have been identified. Number one pattern that we identified is a haplontic type of life cycle. In the haplontic type of life cycle, the gametophytic stage is the dominant stage and the sporophytic stage is the dependent stage or the reduced stage. And where did we study this? In the Thallophyta group, right? In the Thallophyta group, we saw that the gametophyte is the dominant stage and the sporophyte is the dependent stage. So gametophyte in the haplontic life cycle plants is the dominant stage. It is photosynthetic, can make its own food because it is free living, it is multicellular. Whereas the sporophytic stage, it is not free living, it is only one-celled zygote and it is very short-lived. Okay. See, whenever the gametes will fuse, they will form the zygote. Zygote, as in when the zygote forms, zygote represents the sporophytic stage. So, after zygote is not dividing further to form an embryo. The zygote will immediately undergo meiosis in order to form spores. Okay. So, zygote represented the sporophytic generation. Zygote is not forming an embryo. Zygotic meiosis is happening here. And zygotic meiosis, where did we study happened? Zygotic meiosis takes place in many of the algal groups. For example, volvox, Spirogyra, some species of Chlamydomonas. So many algae show this haplontic life cycle. Okay. So this pink colored structure represents the haplontic stage of the life cycle and this blue colored structure represents the sporophytic stage or the diploid part of the life cycle. So you can see sporophytic stage is so reduced and the pink colored stage duration is bigger also and it is dominant also. So this is the gametophyte. Let us look at the life cycle quickly. This is the gametophyte. It will undergo gametogenesis via mitosis in order to form the gametes, which will undergo fertilization or fusion to form a diploid zygote. The zygote represents the sporophytic stage. It will undergo meiosis in order to form haploid spores. As in when the haploid spores are formed, since their ploidy level is in, they come in the haploid part of the life cycle or the gametophytic part of the life cycle because they will germinate via mitosis into the haploid gametophyte. So this was about the haplontic life cycle. And many algae, majority of algae show this kind of life cycle. Okay. Now the second kind of life cycle is the diplontic kind of life cycle. Now in haplontic, the duration of the haploid phase was more, gametophytic stage was more, and the duration of the sporophytic stage was less. In diplontic life cycle, the duration of the opposite is happening here. The duration of the sporophytic stage is higher or bigger and the duration of the gametophytic stage is smaller or reduced. Here what is happening is the gametophyte is reduced and dependent and the sporophyte is free living and independent. And this kind of life cycle is seen in all the gymnosperms and angiosperms because as we are moving down in the plant groups evolution, in the gymnosperms and angiosperms, we are seeing that the gametophyte is becoming dependent and the sporophyte is becoming independent. So the same is happening here. The gametophyte is reduced and dependent on the sporophyte, whereas the sporophyte is independent, free living, dominant, multicellular structure. Whereas the gametophyte is dependent and only single to few celled. Okay, single to few celled if we talk about algae. In gymnosperms, it could be few celled to more than more than a few cells. Okay, so this is seen, this kind of life cycle, diplontic life cycle is see why did I say algae? You must be wondering, because this diplontic life cycle, see, exceptions are always there. So there is one of the algae which is known as fucus, which shows this diplontic life cycle. Although majority of them show the haplontic life cycle, but fucus, this algae, which is also the known as the brown algae, shows the diplontic life cycle. All gymnosperms and angiosperms show this diplontic life cycle, because in gymnosperms and angiosperms, sporophytic is dominant, has a larger duration in their life cycle, in their respective life cycle, and the gametophyte is the reduced stage and the dependent stage. So let us look at the life cycle quickly. See, the blue colored portion is larger in size, pink colored is small in size. So haploid stage is shorter, the diploid stage or the sporophytic stage is larger, the gametophytic stage is reduced. So this is the gametophyte, okay, the gametophytic plant body. It will undergo gametogenesis via mitosis in order to form gametes. The gametes will fuse, fertilization will happen to form a diploid zygote. Zygote will form sporophytic plant body via mitosis. Sporophyte will then undergo meiosis in order to form the haploid spores, which again form a part of the gametophytic generation, These spores will germinate into the gametophyte. Okay, and this is the gametophyte will again undergo gametogenesis via mitosis in order to form gametes. So meiosis is happening where? When the sporophyte is forming, when the sporophytic generation has to switch to the gametophytic generation, only then meiosis is happening. So this was about the diplontic life cycle. Now haplo-diplontic life cycle. In this cycle, both of the stages, haploid stages and diploid stages are present. Okay, these stages are present in all of the life cycles, but what happens here is both are of significant duration. However, this duration can vary. In some plants which follow the haplo-diplontic life cycle, the haploid stage could be the dominant stage, or which occupies the more duration in the life cycle. In some other plant groups which follow the haplo-diplontic life cycle, the diploid stage could be the dominant stage, or which occupies the major part of the life cycle. Okay, so it all depends upon the plant we are referring to, or the plant we are studying. But in general, in haplo-diplontic life cycle, what happens is both the sporophytic stage and the gametophytic stages are multicellular. Up until now, in haplontic stage, we saw that the sporophytic stage was single-celled. The zygote was single-celled sporophytic stage. Now, beyond zygote, meiosis is happening. No multicellular sporophytic stage is seen. And the gametophyte stage here we studied was the multicellular stage. In diplontic life cycle, we saw that the gametophytic stage is the dependent stage, and it was single cell to few celled if we talk about algae, and if we talk about gymnosperms and angiosperms, it could either be few celled or it could be multi-celled, but not we cannot call it as a multicellular structure. Not large number of cells are present, only few cells would be present. And here the sporophyte stage was the multicellular stage. So sporophyte we can call as the multicellular stage because it was a dominant and big structure which we could see properly. But the gametophytic stage was highly reduced. Only few cells would be present in the plants following the diplontic life cycle. But in haplo-diplontic life cycle, both of the sporophytic and gametophytic stages are multicellular. They can be seen well with the naked eye. Okay, we can, like both of these structures, we can call it as multicellular structures because they have that number of cells. They will not be a few cells. Both of these will be proper multicellular structures having the significant number of cells that we need to call a structure as a multicellular structure. So haplo-diplontic life cycle is seen in all bryophytes and pteridophytes. See how the evolution happened. First haplontic life cycle was there. Then haplo-diplontic life cycle came. Then diplontic life cycle. Haplontic life cycle was seen in algae. Then came the bryophytes and pteridophytes, in which haplo-diplontic life cycle came into picture. Then ultimately, what established diplontic life cycle established in gymnosperms and angiosperms. See, an intermediate transition period is happening between in the bryophytes and pteridophytes. See, evolution of the diplontic stage came into the picture in bryophytes and pteridophytes, and this diplontic stage ultimately established itself into the gymnosperms and angiosperms. So, in Haplo-Diplontic life cycle, if we talk about the bryophytes, in bryophytes, the gametophyte is multicellular, dominant, independent, and photosynthetic plant body. Whereas the sporophyte is also multicellular, but it is short-lived, and it is either partially or totally dependent upon the gametophyte. If we talk about pteridophytes, in pteridophytes what happened was the gametophyte was multicellular structure again. It was saprophytic or autotrophic. It was independent and it was short-lived. It was not the dominant structure. See, in bryophytes, the sporophyte was short-lived, whereas in pteridophytes, the gametophytic stage was short-lived. So I told you, depending upon the plant group we are talking about, these stages could differ in their dominance. In bryophytes, the gametophytic stage is dominant, whereas in pteridophytes, the sporophytic stage is dominant. Okay? In bryophytes, the gametophyte was independent and sporophyte was either partially or totally dependent upon the gametophyte. But in pteridophytes, the sporophyte is the multicellular structure, it is the dominant structure, it is the independent, it is photosynthetic and make its own food. So in pteridophytes, both the gametophyte and sporophyte are independent structures, okay? Free living structures, both of these are free living structures. The gametophyte, however, is short-lived in pteridophytes and the sporophyte is the dominant stage. It occupies the major part of the life cycle. The gametophyte part is only is very short-lived. In bryophytes, what did we study? The sporophyte part is short-lived and the gametophytic part is the part that is occupying the major duration of the life cycle because it is the dominant part. So let us look quickly at the life cycle. So haploid gametophyte is there. It will undergo gametogenesis via mitosis. Gametes will fuse, male and female gamete will fuse to form a zygote which represents the sporophyte. It will grow into a multicellular sporophytic structure. Sporophytic structure will undergo meiosis in order to form again spores which represents the haploid generation which will give rise to the gametophyte. Gametophyte will again undergo gametogenesis. So this is how the Haplo-Diplontic life cycle is operating. Here both the sporophyte and gametophytes are multicellular structures, but depending upon whether we are talking about bryophytes or pteridophytes, the duration of these structures can vary. The duration of the duration that they occupy in the life cycle can vary. In bryophytes, the gametophyte occupies the major duration of the life cycle. It is the dominant stage, whereas the sporophyte is the dependent stage. In pteridophytes, what happens is both the gametophyte and the sporophytic stage are independent stages. However, the gametophyte is the short-lived stage and the sporophyte is the dominant stage and occupies the major part of the life cycle. Okay. Now, this is the brief summary, a table that I have made summarizing all the life cycles. You can take a screenshot of this table. You can go through this table. It talks about all the 3 life cycles and the major points and the organisms in which they occur, the plants in which they occur. So, as an exception, we studied that in algae, majority of the algae show haplontic life cycle only, but fucus was an exception which showed diplontic life cycle. Some other algae also, for example, Ectocarpus, Polysiphonia and kelps, they are haplodiplontic. They show the haplodiplontic life cycle. So, algae are showing all the 3 kinds of life cycles, but majority of them have the haplontic life cycle. Okay. Now let's come to the Neet PYQs. This question is from Neet 2023. In angiosperm, the haploid, diploid and triploid structures of a fertilized embryo sac sequentially are cyanogetes, antipodals and polar nuclei, cyanogetes, primary endosperm nucleus and zygote, antipodal cyanogetes and primary endosperm nucleus, cyanogetes, zygote and primary endosperm nucleus. So fertilized embryo sac, if we talk about a fertilized embryo sac, उसमें क्या होता है? What are the haploid structures in a fertilized embryo sac? The fertilized embryo sac haploid structures are the cyanogetes and antipodals. Diploid structure is the zygote that is formed by the fusion of the male and female gamete. Triploid structure is the 3N endosperm, 3N primary endosperm nucleus, or the primary endosperm cell. That is the triploid structure in a fertilized embryo sac. So let us look at the options. Option number 1 is saying that the haploid structure is the synergids. Yes. The diploid structure is the antipodals. No, this is incorrect because antipodals are haploid. So this option is incorrect. The second option is saying that the haploid, diploid and triploid we are talking about. We have to see respectively in the same order these options should occur. That is the first name should be, first element that is being mentioned, it should be haploid, the second part should be diploid, and the third part should be triploid. So the second option, it is saying synergids. Yes, synergids are haploid. Primary endosperm nucleus is triploid, not diploid. So this is incorrect. Again, this option is incorrect. Option number C is saying antipodals. Antipodals, yes, they are haploid. Synergids are also haploid. Primary endosperm nucleus, it is triploid. So this option is also incorrect because synergids are also haploid, but second part of the option should be a diploid structure. So this option is also incorrect. Option number D is saying synergids. Yes, it is haploid. Zygote should be diploid. Yes, zygote is diploid. Primary endosperm nucleus should be a triploid structure. So yes, primary endosperm nucleus is a triploid structure. So yes, this is fulfilling all the requirements of a question. Haploid structure, diploid structure and triploid structure. So yes, this option is the correct answer. Okay, rest all of the three options are incorrect. Let us write the priority levels of the other options also, which we have missed out. Polar nuclei, they are the N plus N structures, and when they fuse together, they form a 2N structure. Antipodals are N. Okay, primary endosperm nucleus is 3N and the zygote is 2N. So N, 2N and 3N are occurring in this order, that is first haploid, second diploid structure and third triploid structure are only occurring in this sequence, and option number D, which gives us our correct answer. Now from Neet 2017, life cycle of ectocarpus and fucus respectively are haplontic, diplontic, diplontic, haplodiplontic, haplodiplontic, diplontic, haplodiplontic, haplontic. So we just studied that in fucus, diplontic life cycle is seen. And in ectocarpus, haplodiplontic life cycle is seen. Both of these are exceptions to the algal groups, right? Because majority of the algae show haplontic life cycle. So the correct answer is respectively it is mentioned. So the first should be haplodiplontic in the options and the second part should be diplontic. So this is seen as option number C only, right? So haplodiplontic and diplontic. So the correct answer is that the life cycle of ectocarpus and fucus respectively are haplodiplontic and diplontic. This is present in option number C. Option number 1, 2 and D are incorrect. So the correct answer is option number C. Now this question is from AIPMT 2014. Male gametophyte with the least number of cells, which means it is highly reduced. Most highly or most reduced is present in. Male gametophyte with the least number of cells is present in Pteris, Funaria, Lilium or Fucus. Now Pteris is a pteridophyte. Funaria is a bryophyte. Fucus is an algae. And Lilium is an angiosperm. Now, how is the evolution in plant groups happening? First is algae, then bryophytes came, then pteridophytes, then gymnosperms and angiosperms. Okay, this is algae and this is angiosperms. Now, as we are moving down from algae to angiosperms, what is happening? The gametophyte is getting reduced. Gametophyte is getting reduced. Here the gametophyte was dominant structure and independent structure. In angiosperms, as we move downwards from algae to angiosperms, the gametophyte is getting reduced and it is becoming dependent. So in angiosperms, the gametophyte is highly reduced and it is dependent upon the sporophyte. So the male gametophyte, if we talk about, is with the least number of cells would be present in which of these plant groups? It would be present, the highly reduced structure with the least number of cells would be present in angiosperms only, right? So the correct answer would be an angiosperm out of these four options, and out of these four options, angiosperm is option number C only, which is lilium. So the correct answer is option number C, because in bryophytes, pteridophytes and algae, in these groups, the gametophyte comparatively has larger number of cells. It is a multicellular structure, or it is an independent structure. But in gymnosperms and angiosperms, finally it is getting reduced up to a stage where it is getting dependent on the sporophyte. Okay, in pteridophytes only we started to see that the gametophyte is getting, the gametophyte was multicellular up until pteridophytes. Okay, in gymnosperms and angiosperms, it became dependent, and it also became highly reduced. Whereas in bryophytes, what was happening was the gametophyte in bryophytes was a dominant structure. Okay, and sporophyte was dependent upon the gametophyte. In algae also the gametophyte was dominant structure and sporophyte was dependent upon gametophyte. In pteridophytes, both the sporophyte and gametophyte were independent structures. In gymnosperms, the gametophyte became dependent upon the sporophyte, and in angiosperms also the same continued. But the gametophyte, which is the most reduced, occurs in angiosperms, because this is the order of evolution that we are following. So the correct answer is option number C. So that was all for today's video, students. Hope you liked the video. Do hit the like button if you like this video. Share this video with your friends, with your batchmates, so that we can reach the maximum number of students. Okay? And if you have any doubts, mention it in the comments below this video and I'll try my best to solve those doubts. Okay, so do let me know if you want a compiled video of all the Neet PYQs from this chapter for the last 14 years as a separate video, a compilation of all the questions that we have covered from the last 14 years. Okay, we have covered all the questions that have come from this chapter in the last 14 years in the Neet exam. At the end of each of my lecture, you can find those Neet PYQs. But if you want a compiled video of the Neet PYQs from the last 14 years for this chapter, I can also post it as a separate video. So let me know of your suggestions, of your thoughts on this in the comment section below if you want them as a separate video for your ease of studying and your convenience. Okay, so that you don't, at the end, you can just look at that video, quickly revise all those questions. If you want that video for that purpose, I'll post that. So let me know about that in the comment section below, and I'll see you in the next video.
Thank you for watching this video. If you have any feedback and suggestions, or if you found something useful, do let me know in the comments below, and like this video, definitely share it with your friends and subscribe to my channel. See you in the next video. Bye-bye.